And then here's some metabolic pathways. Not all of them, but they tried to put them all in one page. And of course, we use this to scare students. Oh yeah, you have to memorize all this. If you look more closely, here's chloroplast over here. Here is mitochondria over here. It's hard for me to see. If I go in a little bit, here's folic acid. If you look over here, I can't even see it myself, to tell you the truth. But what they tried to do is show all the interacting pathways. So right here, glucose 6-phosphate to glyceraldehyde, all the way down to here, to pyruvate. So here is glycolysis. So leading into that would be the Krebs cycle, which is in the mitochondria. And you can follow any intermediate anywhere. This is like a roadmap. So you can say, well, what happens to pyruvate, for example? So here's pyruvate, and it has like traffic arrows. Okay, so you're allowed to go that way. You're allowed to go that way. You can't just go anywhere you want. What I'm really trying to impress upon you is that, yeah, it's impossible to know all this stuff. It's just like impossible to know the entire map of Honolulu, but yet you can use a map to figure out how to get from point A to point B. And that's really the value of metabolic pathways. But also, you know, getting the bird's eye view, you lose detail, but you can see what is the connection between this big metabolic function and this big metabolic function and this one. So this gives you a pretty good idea of what humans have figured out about biology. So anyway, going into the chapter, let me clear some pages here. So what we're looking at here is an overview of metabolism. And so we already talked about the proteome, which just describes all the proteins synthesized by an organism. The connectosome is how different connections are in the neural pathways. The genome is the DNA. Transcriptome is the messenger RNA. Now we can actually talk about the metabolome. It describes all the metabolites found in an organism. So here's generally where we're going. We'll look at bioenergetics. We'll look at one cycle, which is the urea cycle. I guess we'll look at glycolysis as well. Then we go into intermediate metabolism, and then we just go into applications of it. So another thing I want to impress upon you is that the cell has to survive. And no matter what it does, that's its basic premise, is to survive. So when we talked about enzymes not producing any unwanted byproducts, that's similar to what we do in metabolism. You can't have any wasted products. You can't waste energy. You can't at the same time be synthesizing glucose and then at the same time breaking it down to get energy. It doesn't make any sense. You don't go anywhere. It's called a futile cycle. And so this last unit was always meant to be an open book exam. I don't know if I have the right chapter. Yeah, 30s. I think I changed all the chapter numbers. Because I don't want you to have to memorize metabolic pathways. I'm sure you have done it in the past. You have probably memorized glycolysis, memorized the Krebs cycle, but you never even knew what the structures were. You don't understand what you were memorizing. And how long did that memorization last? About an hour after the exam was over, if you ever even got it done. It's truly a waste of time. So I want you to understand metabolic pathways and not just memorize them. So there's two basic categories. Catabolism is the reactions that get energy from food, while anabolism is biosynthesis. Okay? You've heard of anabolic steroids. What do? Well, they make you biosynthesize certain, physiological structures. So here's what life does. So there's CO2 and there's water, and there's energy from sunlight. Plant chloroplasts take that energy from sunlight and those starting products, and they convert it into oxygen and into glucose. Without plants, we wouldn't have oxygen, we wouldn't have glucose. It'd be a pretty boring planet. Then animals, using their mitochondria, and animals in the dark, when they can't use their chloroplast but still need energy, they have mitochondria. They will take the glucose and the oxygen and reverse it back into CO2 and water and give off energy. So how do we control metabolic pathways? Simply by controlling the enzymes. So let's look at some of the players that are involved in making energy. So here's ATP. It's estimated that humans cycle through their own weight of ATP every day. So here's your base, right? This is adenosine ring right here. Here's ribose. Here's a phosphoester. Here's a phosphoanhydride, another phosphoanhydride. So catabolic pathways in the cytoplasm, which is glycolysis, and in the mitochondria, which is the Krebs cycle, oxidize food particles into CO2 and water. And so now I'm not going to use X and XH2 anymore, but this could be an X. That would be XH2. So in addition to this, the presence of oxygen doing the oxidation, this is also an oxidizing agent right here that gets converted into NADH. And so NADH basically has two hydrogen atoms in it. And it has, well, the equivalent of that because it contains the hydride ion. So this is the hydride ion right here in the NADH. It has 2 electrons in it. And so maybe a more correct or more explanatory way to show it is that here's NADH is really NAD plus a hydride ion plus a hydrogen ion. But this is not connected to the molecule, it's just simply in the media. So the actual entity that takes electrons to the electron transport system is the hydride ion. So the hydride ion is a highly reduced form of hydrogen. When you bring that into the electron transport system, then you're reducing the electron transport system with this reducing agent. Okay. And as you'll see, that's what causes hydrogens to be pumped out of the matrix of the mitochondria. When it goes back in, that energy is used to make ATP on the synthesis of the ATP synthetase enzyme. So if you look at NAD, how does it get reduced? What is the actual biochemical mechanism? So here's NAD, and you can see you have 4 bonds on that nitrogen right there. That's why you have the negative charge, the positive charge over here. And when it takes 2 electrons and 1 hydrogen, this is now the reduced form. Okay. So that's a positive, that's a negative. When you put them together, it's neutral, right? Okay. So anyway, some enzymes use NAD, some use NADP. So here is NAD at its full structure right over here. And we can think of why it's called this name, nicotinamide mononucleotide. So here is nicotine. Here it's an amide. This would be nicotinic acid, and now it's an amide. And it's really a nucleotide because of this. This makes it a nucleotide, right? But if you look carefully, it's really a dinucleotide, isn't it? There's two nucleotides there. And so what is the difference between NAD and NADP? It's simply this phosphate, 5, 4, 3, 2 position. Okay, there's a phosphate there. Here is the reduced form, right? So then we have flavin adenine dinucleotide, or FAD. It takes two hydrogen atoms and takes both of them. And here is FADH2. So this is XH2, this is X right here. So here's the structure of it. So here's a flavin right here. So here's the oxidized form. Here's the reduced form. Okay, so this one is a little bit harder to see. Where did the hydrogens go? There's one here and there's one here, both on the nitrogen, right? And this is ribitol over here. You've heard of ribose. That's ribitol, linear form of ribose. And here's two phosphates. That's a phosphoanhydride bond right here. That's why it's called a dinucleotide, because here's one nucleotide. Here's another one over here. So this ribose is circular, this one is linear. But there's enzymes that produce these, right? And here's coenzyme A. So coenzyme A is a pretty long molecule. So, well, what is this? That's adenine. This is adenosine right here. Notice that the phosphate is not on this carbon, it's on the next one. Again, here's a diphosphate right here. So that's the reason why they use the word pyrophosphate, another word for diphosphate. And here's pantoic acid right here, simply a simple acid. So it's an alpha hydroxy carboxylic acid. It's in an amide bond with an amino acid called alanine. But this is beta alanine. So the amino group is not on the alpha, it's on the beta carbon. Then there's another amide over here, and this is ethanolamine. So maybe you remember ethanolamine when we were looking at lipids. And so what is the difference between this and the ethanolamine that's in lipids? Well, this is a thiol instead of an alcohol. So this is mostly abbreviated as coenzyme A-SH. Of course, some people don't put the SH on there. I tend to do that because that makes you understand why does the acetyl group form a covalent bond with coenzyme A. It forms, oh my God, that's a thioester linkage, isn't it? Now you understand why we went through thiols and thioesters. Anyway, so the combination of pantoic acid and beta alanine right here is called vitamin B5. So what if you are deficient in vitamin B5? Well, you wouldn't have enough acetyl coenzyme A. You'd be totally compromised in your ability to produce energy. And then we have coenzyme Q. It's a lipid-soluble redox agent. So here's what coenzyme Q does. Okay. And so here's coenzyme Q. When they're not trying to show you the complete structure, I found one where they show you the complete structure. So you can see why it's lipid-soluble. And you can see that there's a bunch of double bonds in here, but they are not conjugated, so they act like individual double bonds. They don't act like conjugated double bonds. And you can see that this right here is a diketone. And this is essentially on its way to becoming a diol. But the whole thing is aromatic. Okay? That's why it's stable. So how does coenzyme Q accept those two hydrogen atoms? Well, where is it? Okay, this is how it works right here. So you're really reducing two ketones into two alcohols. You reduce a ketone and you get a secondary alcohol. Okay. So mitochondria are the power generators of the cell. They generate about 90% of our total energy. They need oxygen to do that. This is why without oxygen, you don't live very long. And mitochondrial dysfunction is correlated to several different diseases. And as we age, we suffer damage to our mitochondrial DNA. This may reduce the energy production by mitochondria. If you don't keep up with exercise and all that kind of stuff as you get older, you have less and less mitochondria. So in many cases, there are people like myself that will say, well, some people age well and some people age not so well, not so healthy. I think a lot of that has to do with mitochondria. So there's a theory that at one time mitochondria was a small organism that became entrapped in a larger cell. Then they developed a symbiotic relationship. And it's very interesting because if you look at the DNA in mitochondria, it's circular. It doesn't have proofreading or error checking capabilities, so it's more prone to mutation. Mitochondria doesn't get shuffled every generation during sexual reproduction because you essentially get all your mitochondria from your mom. So the sperm tail has most of the mitochondria in the sperm, and it doesn't enter the egg during fertilization. So the human mitochondrial genome has only 37 genes. Most of those are tRNAs. 13 of the proteins of electron transport chain, oxidative phosphorylation. So for example, complex 1 is the membrane-bound enzyme complex that takes NADH and uses it to reduce ubiquinone. So there's 44 proteins in it. 7 of them come from the mitochondria, and the rest of them come from the genome in the cell. In other words, the nucleus. Okay? It's extra mitochondrial. All of complex 2 comes from the nucleus. None of it comes from the mitochondria. You can see there's not that many proteins that are coded for by genes in the mitochondria. So there's a transport system. It's called the TOM-TIM transport system. And it really makes us think that over time, genes from the mitochondria were either lost or transferred to the nuclear genome. So we'll look at the mitochondria. figures right here. So here's a mitochondria. There's an outer membrane. And you see these holes right here are made out of what's called porin protein. And they allow a lot of molecules to go in and out. Not big ones like ribosomes and things like that, but smaller molecules can freely go in and out of the outer membrane. Then the inner membrane is impermeable to most small molecules, and that's where all the enzymes are. Electron transport system or the respiratory chain, the ADP and ATP translocases, ATP synthetase, where you take ADP that's brought in from outside and it's converted back into ATP. This is complex 5 right here. Then if you look at what we're looking at, these big visible dots right here are that huge complex 5 protein that makes ATP. And the inner membrane is what has the electron transport chain, also known as the respiratory chain. And we have these invaginations that go like this that are designed to increase the surface area of the inner mitochondrial membrane so that you can increase the number of respiratory chain complexes that you have. That way, the mitochondria is much more efficient at doing what it needs to do for the organism. So these invaginations are called cristae. Let's see, and here's the matrix. Inside the mitochondria is the matrix. It's totally separate from the cytoplasm. And the matrix contains pyruvate dehydrogenase, citric acid cycle enzymes. So the citric acid cycle occurs in the mitochondrial matrix, as does beta oxidation of fatty acids. And the oxidation of many amino acids happens in the mitochondria. So the mitochondria also has its own DNA, so therefore it has to have also its own ribosomes. It's got a bunch of other enzymes. And here's the porin channels, right, made out of the protein called porin. Here's another view of it from a different textbook that I stole. So you see a slightly different view of it. And this electron micrograph right here, it's unfortunate that this same one is used in almost every textbook. The mitochondria from different tissues look different. Mitochondria that are actively working hard look different than ones that are more in the resting state. So it's a little bit confusing, people. So the outer membrane, you see fatty acid elongation, fatty acid, all this stuff right here is synthesis of fatty acids. So the synthesis of fatty acids happens outside on the outer membrane, essentially in the cytoplasm. But the oxidation of fatty acids occurs in a separate compartment, which is called the matrix. Okay. And this is kind of interesting that the inner mitochondrial membrane is only 30% lipid. Most of that lipid being cardiolipin, that very strange looking diphospholipid, meaning that about 70% of it is made up of all these proteins, especially the electron transport system. Was I not sharing that? Why does that not look like it's being shared?
It was shared.
Okay, thanks. My cursor was on a different spot and it made my green line go away. Okay. What I'm going to point out right here while I have this up is that this is the way they used to show electron transport. And so here's NADH entering complex 1, and there's a big jump right here in energy. You see that 16.6 kilocalories per mole? That was important to them because it takes at least 7.3 kilocalories per mole to make an ATP molecule. So before they really knew what was happening, they knew that there was enough energy over here between NADH and coenzyme Q to make an ATP. But they didn't know exactly how that was happening. Then the same thing over here and the same thing over here. But what I'm trying to get across by showing you this older type of structure is what's really happening over here is protons are being pumped out of the matrix. Protons are being pumped out of here. They basically go into the intermembrane space, which is between the inner and outer membranes. And that's all that happens. ATP is synthesized in a completely different step using complex 5 that is not part of the respiratory chain. So since I have this here, I might as well just talk through it. So here's the intermembrane space. So this is outside the mitochondria. Here's the inside, so it's the matrix, right? So you have to get NADH from outside here into the matrix of the mitochondria. Then that can be used to reduce the electron transport chain. So look at the first thing that's happening right here. NADH is getting oxidized to NAD, and the reducing equivalents, the hydride ion, is given to complex 1. So this is flavin mononucleotide. This is an iron sulfur center. And why is iron important? Because iron can be sitting there in the +3 state. It can gain an electron and turn into the +2 state. So in other words, this protein right here can get reduced. So you're separating the hydrogen ion from the electrons, and the hydrogen ion is pumped out of the mitochondria at complex 1. So notice that it's asymmetrical. So the electrons go this way and the protons go that way. Later on, those protons are going to come back in through complex 5 and make ATP. But they're two separate things. And here's complex 2. So complex 2 doesn't take NADH, it takes FADH2 as its XH2 equivalent, right? So FADH2 gets oxidized, it gives its hydrogen ions, atoms I should say, to succinate dehydrogenase or complex 2. And then you see coenzyme Q, which is a lipid-soluble redox agent, can get reduced at either complex 2 or from complex 1. So this lipid-soluble redox agent, in its reduced form, can then migrate around, and don't forget, 70% of this membrane is protein. There's not a lot of lipids, so it doesn't have to go very far to bump into a complex 3. But when it does bump into a complex 3, it gives its reducing equivalents to the cytochromes in this protein complex. So what happens? 2 more protons go out at complex 3, nothing at complex 2, but you pump protons out at complex 1 and at complex 3. And so the protons go that way. Where do the electrons go? Well, they go to cytochrome C. So cytochrome C also has an iron that's sitting there in its +3 state. And it's a peripheral protein that can kind of move around on the surface, kind of outside the matrix, but closely associated with the matrix right there. And in its reduced form, it will eventually bump into a complex 4 protein and reduce it. Okay, so what happens here? More protons are pumped out of the matrix. This is where oxygen is being used. So the two protons that came in originally over here or over here basically end up making water. Okay. This is where oxygen is being used. So this reduction process right here takes oxygen and reduces it to water. At the same time, you're still pumping protons out of the matrix. So you may be thinking, where are all these protons coming from? Because you only brought 2 in from over here or 2 in from over here, but you're pumping a bunch of protons out. They're coming from the matrix. So what you're really doing when you're pumping protons out of the matrix is that the matrix itself is becoming at a higher pH, right? This is becoming alkaline, and right outside here is becoming acidic. So you set up an electrochemical gradient, similar to what we saw with the nerve cell membrane. You got a bunch of protons out here, and here is going to have a bunch of hydroxide ions. So what would be the motivation for these hydrogen ions that got pumped out to come back in here? Well, the motivation would be there's electrical charge difference across the membrane that you would be able to relax, and the hydroxide ion would be converted into water. So one of the things that never seems to be mentioned when people discuss this and lecture on it is really what drives this to happen is the formation of water. So those protons need a way to get into the matrix to make water out of the hydroxide. They can't come into here because those are one-way pumps. The only way for them to get in there is to go through this portion, the matrix portion, and then, I should say the membrane-bound portion, and then into this part of complex 5. Complex 5 is this entire thing right here. So those protons push through here, causes all kinds of conformational changes, and it essentially takes this ADP and this phosphate and pushes them together so hard that you squeeze out a water molecule and you produce another phosphoanhydride bond. Now you have ATP. So now that ATP has to go out into the cytoplasm again, doesn't it? So you need transport proteins for that to happen. So when I was a graduate student in the early 1980s, I met some of the most prominent scientists of the time that were working on this process. And at that point, somebody had already figured out how this worked, got the Nobel Prize for it. And these people were mad. They were angry old men. Sorry, but they were, because they spent their whole lives trying to figure this out. And somebody came along. He was a rich kid already. So he's got his PhD and he had all this private money. He could just do anything he wanted to. And he figured out how this happened. So tell me, what would happen if I simply punched a hole right through this membrane right here? You got a bunch of protons out here, a bunch of hydroxide ions in here. If I punched a hole right in that membrane, what would happen? It wouldn't go through here because there's resistance. You have to force all these conformational changes to happen, make this happen. They would come right in here, and then you would produce water, but you wouldn't make any ATP. So this is called uncoupling. You're uncoupling the process of pumping protons with electron transport. So you're uncoupling that from the process of producing ATP. And so it's like a simple plumbing kind of explanation that was so simple. They were looking for things that were much more complicated. And yeah, but very, very smart, dedicated people. When I was studying anesthesia, general anesthesia, what I found out is that, you know what all these general anesthetics do? They go to complex 1, they all act at complex 1, and they turn this one-way valve into a two-way valve. So in the presence of, say, any general anesthetic, whether it's inhaled or intravenous, whatever, it binds the complex 1, makes us a two-way channel. You can't make ATP. If you don't have adequate ATP, you can't maintain the electrochemical gradient across your nerve cell membranes. Therefore, they cannot transmit pain and other things. That's another form of uncoupling. So, let's see. Sorry, I'm looking for that other. Covering up all my windows. I can't see where I am. Okay. So here's 3 ways to make ATP. So one is substrate level phosphorylation. That occurs on the surface of an enzyme. So I do not need the mitochondria or the chloroplast to do this. So if I take these 2 reactions right here and I add them together, just like simultaneous algebraic equations, this is the overall reaction I get. So notice that there's 7.3 kilocalories required to do this, but when I do it, I give off 10.3 kilocalories per mole. So the overall is a driving force of 3 kilocalories per mole. So this takes 7.3 kilocalories per mole to do. This one gives off 10.3. So the overall reaction is driven. And what we're saying here is that this endothermic reaction is driven by this exothermic reaction. And this happens on the surface of an enzyme. And then another one is oxidative phosphorylation that we just talked about. So I spelled it out. So NADH comes in, and these are all the same words I was using to explain all that. Okay. And then, oh, here's this. I was thinking, where is that? So here's the outer membrane. Here's the inner membrane. I didn't put a bunch of cristae in there because I just want to make it simple. Electron transport system does what? It pumps protons out of the matrix. What's left in there? Hydroxide, because it's basic in here, acidic out here. It makes it a little bit easier that this acidity could actually equilibrate with the rest of the cytoplasm because of the porin holes over here. But nevertheless, you've created an electrochemical gradient. Here's the electrical gradient. The chemical gradient is that you have a whole bunch of hydrogens out here and a bunch of hydroxides over here. So electron transport system pumps protons out, setting up the electrochemical gradient. The only way for the protons to get back in is through complex 5, the ATP synthetase. And again, what you're really doing is you're producing water. If I put a hole in the membrane right here, I uncouple the process. Electron transport still goes on. I just can't make any ATP. And then the third one is in chloroplast. So, you know, we have several foreign level biochemistry classes at UH, and it's kind of weird because the one that's biomedically orientated, they don't cover chloroplasts, and the one that's agriculture related, they don't really cover mitochondria. But anyway, here's a chloroplast, and you can see the different thylakoids right in there, just like the little units that basically look like mitochondria. So what's going on here? Light comes into the plant. This is called photosystem 1. This is called photosystem 2. So both of those are going to interact with the light. So what does it do? Okay, so photosystem 1, when it's activated by all this energy coming in, essentially takes NADP and reduces it to NADPH. So X gets turned into XH2. You're producing reducing agents. It's the exact opposite of what we're doing with mitochondria. Matter of fact, lots of things are the exact opposite. Now look at photosystem 2. So here's water. You're splitting water inside the lumen, which is analogous to the matrix in the mitochondria. You're splitting water and you're making oxygen. Oh, this is where plants make oxygen. And as the oxygen goes away, it leaves hydrogen. So because of photosystem 2, the inside of the thylakoid, called the lumen, becomes acidic. Now look at, well, this is analogous to the complex 5 in mitochondria, right? Look, it's kind of pointing in the opposite direction, but it's extremely similar. So photosystem 1 creates electrochemical gradient. This is oppositely charged of what goes on in mitochondria. So you have all this protons in here, and they want to get out, they have to go through the ATP synthetase. So hopefully you can see the analogy between mitochondria and chloroplasts. It's the same process that makes ATP. So as the protons go out over here, notice that they come in over here. Oh, look, there's coenzyme Q again. And this is the electron transport system in the thylakoid that ends up giving you NADPH. So this process gives you NADPH, gives you oxygen, and it gives you ATP. So here's a closer look at all that, right? So here's the ATP synthetase. So let me ask you a question. Can chloroplasts become uncoupled? Look at this right here. To get through this ATP synthetase complex, it's not a free ride. You don't just like pass right through. You have to push your way through, cause all kinds of conformational changes in the proteins that squeezes ADP and phosphate together again to make the phosphonhydride bond. If I put a hole in the membrane right here, geez, that's just going to go right through there. And now you have uncoupled light-driven electron transport system from the synthesis of ATP. So both structures need a viable closed membrane to make that process happen. So any questions on any of that?
How long does the electrochemical potential last? Like, does it go through each step and keeps changing, or how does that work?
I'm sorry, the microphone you were talking into, I really couldn't understand the first part of the question. Can you repeat that?
On page 7, you said the phosphorylation of ADP causes the electrochemical potential or consumes the electrochemical potential. So how long does that potential last? Does it build up and then?
Oh, well, you're constantly producing it and constantly consuming it. So in other words, if you stop putting NAD into the mitochondria, the electrochemical potential is not going to last very long.
So it's just a constant process.
Yeah, right. And right now it's not, yeah, it is sharing, okay. My green line is coming and going. But yeah, so what you got to think about in biology is not like some of the chemical processes that actually go to equilibrium. In biology, if you go to equilibrium, you're called dead. So what you're looking at is rates all the time. Okay, everything is based upon a rate. If that rate becomes 0, then you might get to equilibrium, and then that's a problem for you.
Okay, thank you.
So here's the structure of chlorophyll. So you can see how it's going to be anchored in the membrane. It's quite nonpolar over here. And then that looks similar to a heme group, doesn't it? Except it doesn't have iron in there. It has magnesium in there. But this part would be more polar. That part would be nonpolar. And you can see, oh, there's an ester bond right there, carboxylic acid. So you know all these different structures. So now here is the absorption spectra of chlorophyll A and chlorophyll B. Notice that because of these overlap right here of the two different kinds of chlorophyll, this plant can absorb light energy in all these wavelengths over here, all these wavelengths over here. It cannot absorb light at these wavelengths over here very well. So if it's not absorbing that color of light, what's happening to that color of light? It's being reflected. Oh, that's the green part of the spectrum. Now you know why plant leaves are green. One day I'll have time to redo this. Right now I don't. So this is just a generalized view of things. So here's dietary proteins, dietary carbohydrates, dietary lipids, 3 major things that give us food calories, right? So they're broken down, proteins broken down into amino acids. They go into what's called the nitrogen pool, sometimes the amino acid pool. And from there goes off urea. Why? Because you take the amino group off, then the carbon skeletons can be converted into pyruvate or into acetyl coenzyme A or some other type of coenzyme A. The carbon skeletons can go directly into the citric acid cycle. So if you take off the amino group, that's toxic. It has to be converted into urea. And the carbon skeletons can go this way, right? Where all these arrows are pointing to. But notice that an amino acid that you ate could also be turned into a tissue protein, right? And then tissue proteins can actually be converted back and then be used to produce energy. So let's just follow this right here. What if the carbon skeleton was turned into pyruvate? What can happen to pyruvate? Look at this huge one-directional arrow right here. That tells you that most of the pyruvate is going to be converted into acetyl coenzyme A, and you really can't go backwards. Then what happens to acetyl coenzyme A? Well, it can go into the citric acid cycle. It can go through lipogenesis to make new lipid. It can end up in cholesterol and other steroids. It could even go into ketone bodies, which you would really not want to have happen because that's what happens in ketosis in diabetes. But pyruvate can also be converted into glucose by a process called gluconeogenesis to create new glucose. So that's blood glucose right there. So that can be stored as glycogen, or you can go to the pentose phosphate pathway and make NADPH with it. But we'll talk about all the different things that can happen. But I'm just trying to show you an overall viewpoint. So what is this process over here? Because acetyl coenzyme A, which seems to be central to all three of these things right here, it can also go that way. That's lipogenesis, to make new lipid. And here's adipose tissue. So, can you eat protein and get fat? The answer is yes. We just showed you that on this map. Is it the most efficient way to get fat? No, it's not. But the carbon atom from a dietary protein can end up as carbon atom in a fat molecule in a fat cell. Can the carbon atom from a fat molecule end up as a carbon atom in a protein? Okay, well, say we eat lipids, or for whatever, we have fats in our fat tissue. So you go through the spiral over here, which is beta oxidation essentially, and the end product is acetyl coenzyme A. Well, that can go make ketone bodies. That's not helpful. It can't go that way, but it can go down this way. Oh, some of the carbon skeletons from the citric acid cycle can be recycled back into amino acids. So yes, can you eat fat and get muscular? I guess. And of course, eating carbohydrates, can that make you fat? Well, yes, of course. Everybody knows that. So it goes through blood glucose, through glycolysis, to pyruvate, to acetyl coenzyme A, and lipogenesis. Now you're fat. So any questions on this at all? So here's showing you the compartmentalization that happens in organisms. So here's a blood capillary. Here's blood glucose, here's glycerol, here's fatty acids, and of course we talked about these being transported by, different processes, right? LDL, HDL, all those kind of things. So it could be a good health indicator. It could be bad health indicator, depending upon what fatty acids are in here, how much I should say. So here's glucose. It's outside the cell. As soon as it comes into the cell, the very first step in glycolysis is to convert it into glucose 6-phosphate. Now, that glucose 6-phosphate can't leave the cell because it's got that big charged group on it. So it goes into glycolysis, which happens in the cytoplasm. The products of glycolysis are NADH, which then goes to produce ATP in the electron transport system, and pyruvate. Pyruvate goes into the matrix, gets converted into acetyl coenzyme A, and it goes through the citric acid cycle, whose purpose is to make ATP. So here's glycerol. What can glycerol do? Oh, it can go right into glycolysis. Yeah, it can do the whole thing. What about fatty acids? Fatty acids are brought into the cell. They're taken into the matrix, and in the matrix is where the beta oxidation process happens. And the end result of that is to make acetyl coenzyme A, which then goes through the citric acid cycle to make ATP. But what if the cell needs to synthesize some lipids? Well, then acetyl coenzyme A can be scavenged from this catabolic routine, put into an anabolic pathway where you're synthesizing fatty acids. So synthesis of fatty acids occurs in the cytoplasm. Oxidation of fatty acids occurs in a completely different compartment. So you've separated those two processes. It wouldn't make sense for the cell to try to synthesize fatty acids and break down fatty acids in the same cellular compartment. Be too confusing. You probably end up with a futile cycle, just wasting a bunch of energy. So the first cycle we'll look at is the urea cycle. And again, this is not the sequence of events you'll see in another textbook, but I just think this leads more easily into this. So you eat protein, you recycle about 400 grams of protein every day. So blood proteins have a very short half-life, muscle proteins quite a bit longer, and connective tissues way longer. So what happens to amino acids? They're going to be catabolized, because that's what we're looking at in this first chapter right here is catabolism. Well, you take off the amino group, and then the carbon skeleton enters the citric acid cycle, and the nitrogen goes into the urea cycle. So how do we take off the amino group from an amino acid? Well, transamination. And notice that alpha ketoglutarate. What is that? Here's the alpha carbon. Oh, it's a keto. And this is glutamic acid. Okay, it's thioic acid. So anyway, the amino group gets transposed from here to here. So now you're making glutamic acid. Oh, that's a normal amino acid, isn't it? So we're taking any amino acid and we're turning it into a specific amino acid called glutamic acid. And that's done by this enzyme right here. So this alpha keto acid can then enter the Krebs cycle known as the citric acid cycle. So that's one way. Another way that occurs for glutamic acid is oxidative deamination of glutamic acid by a different enzyme. So, alpha ketoglutarate and alpha ketoglutarate. In other words, what happens to the glutamic acid? Like whatever incoming amino acid comes in, it gets its amino group given to alpha ketoglutarate. So they all end up as glutamic acid. So what happens to the glutamic acid? Another enzyme turns it back into alpha ketoglutarate, and you give off free ammonia. So in some organisms it uses NAD, some organisms it uses NADP. So the process is complicated because this right here is, well, we call it oxidative, but at the same time we've got to reduce something, right? This is what you're reducing. NAD goes into NADH. So glutamate, glutamic acid, central to this process, isn't it? Very important. Then finally we have the direct deamination of serine and threonine. So serine and threonine differ only by this methyl group right here. So serine is just this, right? Threonine would be with that methyl group right there. So this enzyme simply directly takes off that amino group right there. This can actually go into the citric acid cycle. So what about the urea cycle? We just now showed you why you need the urea cycle, because ammonia is toxic. We talked about that in many different places in the course. So how do you get rid of that ammonia because it's so toxic? Well, the overall process takes two ammonia molecules, a CO2 molecule, uses some ATP, and you make urea and water. Okay, so you're using a bunch of ATP over here, but I got to show you with the cycle, it's much more efficient than that. So this is not basic, this is basic. That's why this is much less toxic. Okay, so let's see what happens. Here's inside the matrix of the mitochondria. Here's in the cytoplasm. Okay. So let's see, where's a good place to start? So here's where you actually create the urea, right? So here's CO2 and the ammonium ion. So why did I take ammonia and make it into the ammonium ion? Because in the matrix it's quite acidic, isn't it? Because of electron transport. So we spend two ATPs to make a very high energy compound called carbonyl phosphate. It's high energy because if you look at this, this is what's called a mixed anhydride. That's an anhydride between phosphoric acid and a carboxylic acid. Okay? And it took a lot of energy to do that. But the reason we're willing to spend this energy is because ammonia is toxic. So carbonyl phosphate is a high energy compound. It's only found in the matrix where it's made. And then there's another amino acid right here. Here's the acid, here's the amino group, right? So ornithine is an amino acid not found in proteins. It's brought in through the matrix, into the matrix where carbonyl phosphate is, and they're combined. And then you get an amino acid called citrulline. So citrulline can be formed in the matrix, come out, and go into the cytoplasm. It's getting to be a pretty large molecule, isn't it? So then aspartic acid comes in, is combined with citrulline to make this bigger amino acid, argininosuccinate. And look, you're using more ATP to do that. So here's the amino acid. Right now, look at what's going on right here. You see how this is starting to look pretty weird, right? Remember the amino acid arginine? Well, that's really what we're going to be doing. We're going to convert these two amino acids into this amino acid, take off some carbon compounds, and basically generate arginine, which is a normal amino acid found in proteins, but it's also involved in the cycle. So then what happens to arginine? Well, you can see right here, it's like there's so many weird nitrogens on there. I wouldn't even be surprised if something like ammonia comes off. When you take the ammonia off right here, you regenerate ornithine, which then continues the cycle. So what would happen if I ran out of ornithine? Well, you can make it again, right, from aspartic acid. But you can see if you ran out of any of these things, the whole thing would stop. But it's not likely that you would run out of any of these things. If your mitochondria ran out of energy, it wouldn't be able to produce ATP, you wouldn't be able to produce carbamoyl phosphate, you wouldn't be able to carry out the urea cycle. Now, the result is that we just produce a molecule of fumarate. This is fumaric acid. It's a dioic acid with a double bond in it. That enters the citric acid cycle and gives you 3 ATPs. So this is not the whole story over here, because even though if you look at it in this point, fumarate goes into citric acid cycle and produces 3 more ATP. What that really means is that it takes only 1 molecule of ATP to make a urea molecule. And that 1 molecule of ATP gets rid of 2 ammonias. So I put some information like that on here. Let's see. There's 5 intermediates that are unique to the urea cycle. Some are in the mitochondria, like carbamoyl phosphate. Others are both, right? They got to go in and out. So there has to be shuttles for that, transport proteins. Okay. So anyway, that's your first cycle. Any questions about it? What about glycolysis? So this happens in the cytoplasm. So all the enzymes are in the cytoplasm. And here's the overall pathway for it. So glucose has to be oxidized. So here's a 6-carbon carbohydrate. It's being oxidized into 2 3-carbon molecules that are oxidized. And I have 2 ADPs that are making ATPs. But you see this NADH right there? This is before the electron transport system happens. This is without involving the mitochondria in the use of oxygen. So the product of glycolysis is pyruvate, NADH, and ATP. Later on, this NADH can go into the matrix and go through electron transport system and make more ATP. But right now, we're looking at just what happens in the cytoplasm. So let's look at this. And again, the whole purpose of oxidizing glucose is to make energy. So the first step is this. Here's glucose. An enzyme called hexokinase takes an ATP molecule and phosphorylates it. Like, what? This is illogical. If glucose is supposed to give you ATP, why are you already spending an ATP in the very first step? It just so happens that glucose 6-phosphate now cannot leave the cell. It's trapped in the cell. So that's kind of a good thing. But I call it a setup reaction. Okay, so let's look at the next step. So glucose 6-phosphate gets isomerized into fructose 6-phosphate. It's like, what? You turned an aldehyde into a ketone. The reason why it's not obvious is because they're showing you both in the Haworth projections, right? So no atoms were gained or lost. You simply converted an aldehyde to a ketone and then put it back into a cyclic form again. That's why it's called an isomerase, phosphoglucoisomerase. It certainly won't isomerate this, but it will this. So anyway, that costs us hardly any energy right there. And then we get to the next step. So fructose 6-phosphate is, oh my God, you're spending another ATP? That doesn't make sense. But again, I call it a setup reaction. So now you have a molecule of fructose 1,6-diphosphate, also known as bisphosphate. And what you've done is made a molecule that is almost symmetrical. Not perfectly, but almost symmetrical. Because the purpose of the next step of aldolase is to split it into 2 3-carbon molecules. So if you did it here or here, it wouldn't be symmetrical, but now it's symmetrical. Both of these 3-carbon molecules have phosphate on there. So notice that they're showing you this going into the next step. So this 3-carbon, which is glyceraldehyde 3-phosphate, goes into the next step, but this one doesn't. But you cannot waste anything in a metabolic pathway. So what happens is a dihydroxyacetone phosphate. Now, if you look at the name right here, I'm opposed to using these old-fashioned names. But look, carbon, carbonyl, carbon, that's acetone. Dihydroxy means you have a hydroxy on number 3, hydroxy on number 1. And then you put a phosphate on it. And a phosphoester linkage right there. Well, anyway, it takes not much energy at all to convert dihydroxyacetone into glyceraldehyde 3-phosphate. So you essentially took the 6-carbon and you have 2 molecules, each with 3 carbons, and they're both glyceraldehyde 3-phosphate. So from here on, everything could really be doubled. Okay, so you make 2 pyruvates, 2 phosphoenolpyruvates, right? You make 2 NADHs. If we look at the next step, what's going on here? Glyceraldehyde phosphate dehydrogenase. A dehydrogenase is the old word for an oxidoreductase. So this enzyme has to bind glyceraldehyde 3-phosphate. It has to bind NAD, and later on it has to bind also phosphate, and it converts NAD into NADH. Well, what happens to the NADH? Goes into the mitochondria through the electron transport system and makes ATP. So here my setup reactions required 2 ATPs for 1 glucose molecule. Now I came over here, and each one of these is going to produce an NADH. So I got 2 NADHs. Every NADH is going to give you the equivalent of 3 ATPs. So I'm already well ahead. I spent 2. Now I got 6. I'm ahead 4. And I still got 2 molecules of 1,3-diphosphoglycerate. So this is an anhydride. Okay. And so look what this next enzyme does. It binds 1,3-diphosphoglycerate. It binds ADP. And what does it do? It takes the phosphate here and transfers it to ATP. I am making ATP not in the matrix, not using the electron transport system, but it's happening on the surface of this enzyme right here. That's substrate level phosphorylation right there. When NADH goes into the mitochondria, that's called oxidative phosphorylation. This happened substrate level. So I made 2 more ATPs because I got 2 more of these molecules, right? And I still got 2 molecules of 3-phosphoglycerate, which goes through an isomerase. Mutase is the same thing as an isomerase. Now I got 2 molecules of 2-phosphoglycerate. And then the next enzyme called enolase. What is it doing? It's taking this 2-diphosphoglycerate and dehydrating it. Where is the water coming from? Well, here's the OH right here, and here's the H right here, because I'm making that double bond right there. And it doesn't take very much energy to do it. And now I have 2 molecules of what I call PEP, phosphoenolpyruvate. So here's phospho, all that would be, right? If I didn't have the phosphate, it would be an enol. And enols are already very reactive. When I put the phosphate right there, it's still very reactive. And so the double bond here and the double bond there and the double bonds here, there's simply a lot of double bonds in there, a lot of repulsion in that molecule. So pyruvate kinase binds molecule of PEP, molecule of ADP, and directly transfers the phosphate to the ADP. Guess what? I just made 2 more molecules of ATP. And I still have 2 molecules of phosphate left over. Notice that we didn't make any carbon dioxide. 6 carbons came in. We still got 6 carbons, right? I'm getting ATP from substrate level phosphorylation, and I'm getting ATP if that NADH goes to the matrix of the mitochondria and makes ATP by oxidative phosphorylation. So what I want you to understand is the strategy of these pathways. It's just amazing, actually. I remember my father being a businessman. He was a construction contractor here in Hawaii. And he used to always tell me, oh, you have to spend money to make money. Turns out to be true. So we know what allosteric effectors are. Here's a control point. Here's another control point over here. Okay. What controls this enzyme right here? This is an allosteric enzyme. It's turned off by ATP, turned off by citrate. Why would that be the case? If the purpose is to make ATP, and I already have a whole bunch of ATP in the cell, why am I going to keep trying to make ATP? I can use that glucose for something else. Same with citrate. Citrate is the first intermediate in the citric acid cycle. If I'm not using that, the citric acid cycle is off. That's the same mentality as having a bunch of ATP. I don't need to make more citric acid. So it makes sense to turn off glycolysis. That means that this will build up, that will build up, that will inhibit this enzyme. And eventually, glucose will stop coming into the cell because that cell doesn't need it. That cell doesn't need it, and it doesn't take the glucose, and that glucose can continue along this pathway in the bloodstream and go to a cell or a tissue where it is needed. So really, this is a way for a cell to act like it's part of a multicellular organism. And what would it mean to a cell if I had a whole bunch of AMP? Well, if I have a whole bunch of ADP, that means I'm going to have a whole bunch of AMP. So that means, like, you know what? We're in a situation here where we need a bunch of energy. So we got to turn these things on and make energy. Then what about down here? Glucagon, cyclic AMP, at least in the liver, turns this system off. So glucagon is the exact opposite of insulin, right? So insulin will turn this system on. Glucagon would have the exact opposite effect. So now you're letting your liver control the enzymes in these cells, especially in liver cells, to help regulate blood glucose levels. And when we get to fructose 2,6-diphosphate, it'll take a little bit more of an explanation to explain how that regulates. So I went through step by step. Okay, anyway. So let's look at the energy production and glycolysis. So here's step by step, right? And if I have oxygen and I can take all my NADH, turn it into ATP, I can make 6 ATPs in skeletal muscle from 1 glucose molecule. In heart and liver tissue, I can make more. That means that heart and liver tissue is more efficient than skeletal muscle. We'll see why that is, because it costs energy to get NADH into the mitochondria in skeletal muscle. But most likely the reason why you have that inefficiency built in is because inefficiency generates heat. You need heat in your muscles to keep them ready for action at any time. Then what happens without oxygen? Well, you can only make ATP by substrate level phosphorylation. So anyway, so if you look without oxygen, right, which is in blue right here, take out the blue part, here's your 2 setup reactions. You're making 2 there and 2 there, but those 2 subtract from one of those 2, so you only get 2 without oxygen. Okay. Well, I guess that ends it right there. I saw this cartoon one time and I go, yeah, that's exactly how I felt as a student. Exactly how all biochemistry feels as a student. And so why do we keep perpetuating it? So anyway, on exam 5, you will have all this available to you. You do not have to memorize any pathways, but I will be asking you questions about the pathways. So you look at the pathways and try best to answer your questions. Okay, so any questions so far?
On exam 5, on the syllabus, you said Monday is possible. Do you know when you're gonna tell us if we'll have until Monday for it, or is it definitely gonna be on Friday?
No, I'm pretty sure it'll be on Monday. I probably should recheck after I've rechecked many, many times, because they constantly send us emails updating stuff. And it's like, it's essentially when our grades due. It's really what it means. So for me, and theoretically in a face-to-face class, you got 6 weeks, and then the last day of school is your final, and you just got lectured on it the day before. I think that's just ridiculous. Anyway, so yeah, most likely Monday is possible for the exam.
Okay, thank you.
Okay, so we're not going to lecture anymore, but I just wanted to show you where's my sharing? What the next chapter looks like. Okay. So intermediate metabolism. So here's what happens to pyruvate, right? So we're going to look at all the different things that happens to pyruvate after it's made in the cytoplasm. And then we're going to look at some other things like making glucose. So glycolysis versus making glucose. And then we get into some regulatory things, making glycogen, more regulatory things. Then how do we get NADH into the mitochondria? And then the citric acid cycle. Okay, so that's what we'll be doing tomorrow. Okay, so the citric acid cycle might look very, very complex, but again, this is going to be right in front of you for the final exam, and I'm simply going to ask you questions about it. And there is a strategy that the citric acid cycle uses, and that's the kind of thing that I'm interested in. Okay, so any questions before we take a break for the day? If not, I'll have this up on YouTube. I don't know, doesn't take that long. Sometimes it takes longer than other times, but it should be within an hour or two. If you got other questions, just email me and I'll let you know. Somebody's asking how many questions there are in how many chapters in Unit 5, where there's these two, and then there's one on disease and there's one on nutrition, which I doubt if we get through them, but those are basically applications of everything. And they're more geared towards making you be able to find something you're interested in to do one of the extra credit papers. That's kind of the idea with those. Okay. So hopefully I will see you folks tomorrow. You guys have a great day.
Thank you, you too.
See you.